this bacteria Salmonella and how it takes a journey from contaminated food into our body in order to cause food poisoning. And today this talk is going to be broken up into 4 different parts. First, I'm going to be talking about how food poisoning is a type of infectious disease. Next, I'll be describing the bacteria Salmonella. And then Brandon will take over and talk about the first part of Salmonella's journey, how it travels from food into our body and into our intestine. Next, Greg will take over, last but not least, to talk about how once Salmonella is in our intestine, how it makes a home for itself there. And so first up, food poisoning is actually a type of infectious disease. And I think we hear about food poisoning a lot in the news. For example, you can hear about outbreaks and recalls, and there's been a couple of really notable examples of food poisoning here in the Boston area and in surrounding states over the past few years. And especially this norovirus outbreak in Boston this time last year. I hope none of you guys, were unfortunate enough to go to Chipotle last November because over 100 people were sickened from this outbreak. The popular restaurant here, Clover, shut down briefly a few years ago for a Salmonella outbreak. And then this summer, there was a recall of frozen food from Trader Joe's due to listeria contamination. And there was another Salmonella outbreak in 9 states that was linked to alfalfa sprouts. So I want to highlight these 2 headlines. And the first says, the US sees hundreds of foodborne illness outbreaks each year. So these types of outbreaks are pretty widespread. They're impacting people across the US. And then this last headline refers to the Chipotle outbreak as a scare. And when this type of foodborne illness outbreak is happening, it is really scary. And I think, so as microbiologists, we kind of want to talk today a little bit about the bacteria that are behind these foodborne outbreaks. So maybe next time if you see a headline kind of like this, you can understand a little bit better what's actually happening inside our body. And food poisoning for a healthy adult usually looks something like this. Here you are, happy living your life, and if you, were unfortunate enough to go to Chipotle last year, a few days later, be feeling really sad, maybe experiencing some nausea, some diarrhea. And I think for a lot of people, we think, okay, I ate this burrito, it was the burrito that made me sick. But it's actually not the food that's giving you food poisoning, it's the germs that are on the food. And today we're going to be talking about a specific type of food poisoning caused by Salmonella that's known as diarrheal disease. And this just simply is called that because the main symptom is diarrhea. Food poisoning is a type of infectious disease, and infectious disease can be defined as an illness that is spread by a germ from person to person. And I think an example that we've all kind of thought about is, say, the cold or the flu. Here's a person on the left. Imagine that these red ovals represent the flu virus or the flu germ. And the person on the right is healthy. If you're coughing and sneezing, and you happen to cough on someone, you can pass this germ from your body onto their body and make them sick. And I think this is something that we kind of intuitively understand, but this is actually also true, for food poisoning as well. If you have diarrhea caused by a germ from food poisoning, and if you say don't wash your hands very well, the next person you shake hands with can then get the same diarrhea without ever having eaten that contaminated food. And infectious disease actually is really important worldwide. It kills a lot of people around the globe. So if we look at this pie chart of the major causes of deaths worldwide each year, infection accounts for almost 18% of deaths. And this is about almost 10 million deaths per year, which is a pretty big number. And this is actually, a huge problem for specific populations of people, especially for low-income countries where infection is actually the #1 cause of death. And also for children under the age of 5, elderly, and those with weakened immune systems, they are really susceptible to not only acquiring infection, but also from dying from them. So this is a really important problem, and we are interested in learning about this so we can help, understand it better to maybe develop new treatments and cures. So if we break down this slice of the pie that corresponds to infectious disease into all of the different types of infections that you can get, There's a few major ones that pop out. So pneumonia and tuberculosis are both infections of the lung. HIV and AIDS is a disease that impacts the immune system. And today we're going to be talking about diarrheal disease, and that accounts for 15% of all of the deaths attributable to infection. And so that comes out to be about 1.5 million deaths each year. And thinking even more broadly, 1.7 billion cases of diarrheal disease each year. So of all those billions of cases, 1.5 million people will die, but that's still millions and millions of people experiencing diarrhea, which, obviously is unpleasant. And if we can do anything to kind of stop this public health burden, we would really like to be able to do that. And I mentioned that infectious diseases are caused by germs. So what are germs really? They're simply microscopic organisms that cause disease. And this phrase, microscopic organisms, is kind of a mouthful, so we usually just refer to them as microbes. And as a reminder, microscopic just means you can't see it with your eyes. You need to use a microscope. And here is a photo of our very own Brandon using a microscope in our lab to look at some cells. And here's a picture of what he could be looking at. These are some human cells on a grid, and that allows him to count the number of cells that he has in his sample. And so this microscope is actually relatively low powered, and there's even higher powered microscopes that we can use to study microorganisms in even greater detail. And broadly speaking, there are a few different categories of microorganisms, and these include viruses, bacteria, protozoa, and fungi. And in order to really understand the next part of the talk, when Brandon and Greg are going to be getting kind of into the more complicated details of a Salmonella infection, I want to pause here for a few minutes and talk about some basics of microbiology, so as we move forward, we can really understand what they're talking about. So, as I just said, microbiology is the study of microscopic organisms, and there are different categories of microbes, and here are some cartoons of them. Again, we have viruses, bacteria, fungi, and protozoa. And there are many members within each of these categories. So there are many different types of viruses, there are many different types of bacteria, and the same is true for fungi and protozoa. And an important point I want to impress upon you guys is that most of these members do no harm, and in a lot of cases they are actually really good for us, and only a very few of them have the ability to cause disease, and those are the ones that we refer to as germs. And I think two questions, can arise from thinking about these microscopic organisms, and that is, how small are these actually compared to what we can see in our day-to-day life, and where do they live? And so to address this first question of how small they really are, I want to kind of zoom way out the other end of the spectrum and talk about the largest animal on Earth, and that is a blue whale. And a blue whale measures 25 meters, that's about 80 feet from end to end, which is about 15 times longer than an average human, which on average come in under 2 meters tall, or about 5'5". And so you can see this diver is drawn to scale. So the blue whale, like I said, is about 15 times longer. And kind of on the other end of the spectrum is a mouse. And this is something that we, coming in only a few inches long, we might consider to be quite tiny, but compared to the microscopic world, mice are actually ginormous. And so mice, humans, and blue whales, what they all have in common is they're built of animal cells. And here's a cartoon of an animal cell. And a cell is just a microscopic building block of a living thing. So all animals are made up of animal cells, plants made up of plant cells, and so on. And animal cells are about 25 micrometers across, and a micrometer is 1/1,000,000th of a meter. And that's kind of hard to wrap your brain around how tiny that is, but if you think of it that there are 30 trillion cells in the average human's body, it's pretty amazing how tiny these guys actually are. And so if we can dive down to this level into the microscopic world, I can bring up some of those microbes that I just mentioned and see how they compare to the cells in our own body. So animal cells, again, are coming in at about 25 micrometers in diameter. Fungi, this is an example of a yeast cell, are 5 micrometers across, and as some of you guys might know, we really like yeast because they're what's responsible for, making bread and beer and wine. So some of our favorite microorganisms. And we also have protozoa, which are about 4 times as long as animal cells. And these type of microbes can be found swimming in water and also in soil. And what all these different cell types have in common is that for cells, relative speaking, they are all quite large and they are pretty complex. So if you look at this cartoon of an animal cell, it has lots of different, subcompartments, and these subcompartments all help the cell, do its cellular functions. But today we're going to be talking about bacteria, and bacteria are actually even smaller than those cells that I just described. And here is a cartoon of a bacteria. From end to end, they're measuring just about 1 to 2 micrometers in length. And again, a micrometer is 1/1,000,000th of a meter. So these guys are incredibly tiny. Here's a picture of a bacteria, roughly to scale with a human cell. And the size difference between a bacteria and a cell in your body is about the same size difference between a human and a blue whale. So it's pretty amazing to think, despite how tiny bacteria are, they're able to enter our bodies and actually mess things up that are going, the way that they're not supposed to be. And the last type of microbe that I'm going to talk about today is a virus. And viruses are incredibly tiny. They are about 20 times smaller than a bacteria. And again, this size difference is the same difference as a human and a blue whale. And viruses, as an interesting fact, is that they're not considered to be cells, so we refer to viruses as particles. And just to summarize what I just said, so humans, like other animals on Earth, are made up of animal cells. And there's a whole world of diverse different types of microorganisms, namely bacteria, protozoa, viruses, and fungi. And to address the second question that I brought up in the beginning of this section is, where do these microorganisms live? And I want to impress the point that we are really living in a microbial world. So microorganisms exist on every single environment on Earth, including ones that would be completely inhospitable to humans, like, incredibly hot springs. The water would just absolutely be too hot for us to live in, as well as extremely cold temperatures. There are bacteria there that are just thriving. And there are also bacteria that make their home in our bodies. And there's 2, main areas on our body that bacteria find home, and that is on our skin and in our intestinal tract. And again, I want to really reiterate this point that most of these microorganisms that exist on Earth are good. They are helping our planet's ecosystems, and they're actually helping our health. For example, some microbes that reside in our stomachs and in our intestine can actually help our immune systems learn how to fight off the bad bacteria that we don't want in our body. And very few of them cause disease. And I want to share this really cool quote by scientist Dr. Carl Woese, who's a pretty famous microbiologist. He said, "If you wiped all multicellular life forms off the face of the earth, microbial life might shift a tiny bit. If microbial life were to disappear, that would be it, instant death for the planet." So hopefully this quote can impress upon you that these guys are really important, not just for our health, but for the health of the planet. And so I just told you that microbes can live in any environment on Earth that they want. So what do they want with us? Why do they want to live in our body? And I think this can be illustrated with another quote from another famous scientist. Francois Jacob said, "The dream of every cell is to become 2 cells." So here's a bacteria, and all it wants to do in its life is settle down, find a nice home, and make more of itself, not unlike us humans. And they're just trying to find a good place to do this. And sometimes the best place for a bacteria or any other type of microbe to do this is inside our body. And a lot of times, like I said, this can be beneficial to us or doesn't cause us any problems. But sometimes when those microbes find a nice home in our body, that's when we have an infectious disease. So I want to pause here for a summary. I just told you that food poisoning is an important infectious disease. Infectious diseases are caused by microscopic organisms and are spread from person to person. And bacteria are single-celled organisms that can be both good and bad for your health. And I can take any questions now before moving on to the next part. Yeah?
Is Salmonella the only bacteria that causes food poisoning?
The question was, is Salmonella the only bacteria that causes food poisoning? And no, there's actually a lot of different ones. One that comes up in the news a lot is E. coli can also cause food poisoning. And there's some like cholera that happen, not so much in the US, but in other countries. Other questions? Okay. So next up, I'm going to talk specifically about the bacteria Salmonella. And in the first section, I told you, diarrheal disease is a really important infectious disease. So like any infectious disease, diarrhea is caused by microorganisms. And to show a few examples, the different types of microbes that can cause diarrheal disease, we're going to go through different types and name an example of each. So first up, we have viruses. An example of that is norovirus. That's what caused the Chipotle outbreak. And here is a picture taken on a really, really high-powered microscope of norovirus particles. And this image has been colored blue. Another example of that is protozoa. And one member of this category that can cause diarrheal disease is Giardia. And these are like these little monsters, and so remember, they're microscopic, so you can't see them unless you're looking under a microscope, but these guys are actually attached to the intestinal wall in this image, and again, this is taken with a really high-powered microscope. So, you don't want this in your intestine. And lastly, the topic of our talk today is bacteria. And we're going to be talking all about Salmonella, but here's another really cool image taken with a high-powered microscope of bacteria. And I want to introduce this term here that you'll hear Greg and Brandon talk or use in their section, and that is the word pathogen. And pathogen is just another word that means germ. So, norovirus, Giardia, Salmonella, and other germs can be referred to as pathogen, which just means a microorganism that can cause disease. And a little bit about the bacteria Salmonella that we'll be talking about today. It's this long rod-shaped bacteria, and it has these appendages sticking out, which are called flagella, and they are used they're used by the bacteria to swim through our body. And some stats about Salmonella infection. It's a really major source of food poisoning around the world. It causes 94 million cases, resulting in 160,000 deaths each year. In the US alone, it causes 1.2 million cases of food poisoning, resulting in 450 deaths per year. And again, these deaths and cases are more often than not happening to children and elderly and other people who may have a weaker immune system. And I want to go through, what happens to you if you're unfortunate enough to encounter Salmonella and it enters your body. So again, happy, healthy adult, totally unsuspecting, and somehow you get exposed to the bacteria, usually by eating something, and then within 3 days, you're going to be feeling really sick because the Salmonella has entered your body and is causing diarrhea. And the main symptoms you experience are diarrhea, fever, and cramps. And now for most healthy adults, the way we treat this is just by rehydrating to replace the fluids that you lose from the diarrhea, and then in about a week, you're feeling better. So antibiotics are not even routinely used unless the infection is really, really bad. But the way people die from Salmonella infection is that they lose so much water from the diarrhea that they become dehydrated, and that's the cause of their death. So again, children, elderly people who have, it's easier for them to get dehydrated. That's the reason why they are going to be dying from this. And also, like I said before, low-income countries where they might not necessarily have a good source of water or clean water, it's going to be hard to replenish those fluids, so they can unfortunately die from this disease. And here in the US, we keep track of infections like Salmonella, and the Center for Disease Control kind of keeps logs of what did people, how did people get this type of infection. So if we look over the last 5 years of all the ways that people in the US got Salmonella, hopefully we can see some trends popping out. So in 2016, this year, it was reported that people got Salmonella infection from sprouts and chicken. In 2015, they were infected by turtles, cucumbers, pork, chicken, and geckos. In 2014, cucumbers, sprouts, chicken, and bearded dragons. In 2013, poultry, cucumbers, and eggs. And in 2012, hedgehogs, cantaloupe, poultry, ground beef, and eggs. So let's kind of start with the ones that might make more sense. And I think what a lot of people might have heard is that Salmonella exists in chicken products. And you can get Salmonella from eating chicken breast or meat from the chicken, as well as from eggs. And the Salmonella exists on these types of food, but if you cook the food, you're killing the bacteria. So that's why you've probably heard, don't eat raw cookie dough because you'll get food poisoning, and that's from Salmonella. And so there's some things on this list that might not intuitively make sense, like bearded dragons. And I think this illustrates a really good point that Salmonella actually exists in the intestines of tons of different types of animals. So it exists in birds like parrots, parakeets, poultry, as I just told you, chicken is a really big source of Salmonella. Reptiles, of course, here is a picture of a bearded dragon, as well as turtles and lizards. And our household pets like dogs and cats, livestock, and frogs. And so if Salmonella is in the intestine of these animals and we are handling those animals, the bacteria can be passed to our body. So there's really 3 main ways that you can get Salmonella infection. That's from eating contaminated food. So if there is bacteria in the intestine of these animals and the animal is slaughtered to prepare the meat, the bacteria can get on the meat, and when we eat it, we get sick. For vegetables, this is usually caused by some type of cross-contamination, either out in the field, if, for example, the cow is shedding feces that contains diarrhea, and somehow the water is bringing it over to the vegetables. That can be another way that it's contaminated. You can get Salmonella from directly handling these infected animals. And lastly, as I mentioned before, you can spread your diarrheal disease to your friend if you don't wash your hands. And again, I want to stop here for a summary and answer any questions. So I just told you that viruses, protozoa, and bacteria can cause diarrhea. And today we're talking about Salmonella. Diarrhea from Salmonella is usually short-lived for healthy adults, but it can be deadly to some. And exposure is caused in 3 ways: by eating contaminated food, handling an infected animal directly, or human-to-human spread. So if anyone has any questions about this part, yeah.
You are talking about we can get sick of having this bacteria, but I never listened any kind of dog dying of Salmonella or a cow dying of Salmonella. So does this affect different the human beings and the animals, or how that works?
So the question was, does Salmonella infection affect animals differently than it does humans? Because we don't really hear about, how often do you hear about your dog getting Salmonella? Well, yes, it does affect humans and animals differently. We know that Salmonella can exist in some of these animals, but a lot of times, we're not even really sure if it's making them sick. I think it's something that's not really well understood or well studied, but we do know that if the Salmonella is present in the animal, it can be passed to us, and then once it's on our body, it makes us sick. Hopefully that answers your question. Yeah.
Is salmonella always in us? So like right now, is there some of that bacteria in you and it's just sometimes it causes the disease and sometimes it doesn't?
So the question is salmonella always in our body and then sometimes it's causing infection and sometimes it's not? And the answer to that is no. When it's in our body, it will give you food poisoning. But that is, there are bacteria that exist in our intestine naturally and we want them to be there, and those ones are not disease causing. Yeah.
There are bound to be variations in sensitivity to salmonella, but within one person, can one person develop a sort of increased resistance to the ill effects of salmonella?
So the question was, can one person develop resistance to salmonella? So if you get infected and you encounter it again in your life, you won't, you're asking, will they be infected again? And the answer for that is actually, yes, you can be reinfected because there are actually a lot of different subtypes of salmonella, and the chance of you encountering the same subtype twice is pretty rare. So if you were to encounter the same subtype, then potentially immunity in your own body could develop, but the chance of that actually happening is pretty low. So if you're seeing it again, you're gonna get sick again. Yeah, that's okay.
That is the reason because there is not a vaccine against salmonella. It's because of different kind of salmonellas. For example, you talk about the TB, right? We have vaccines for the TB, but we don't have vaccine for the salmonella.
Right, so the question is there a vaccine to salmonella? And the answer is no. And was that your only question? Why is there no vaccine? So I think that's a really good question, and a lot of labs here at Harvard and at other medical schools around the country are working on vaccines for diarrheal pathogens like salmonella. For example, in my lab, we're working on a vaccine for another diarrheal disease, cholera. So some of these vaccines just haven't been developed yet. Yeah.
Fun fact, salmonella can actually affect dogs occasionally and cause diarrheal disease, but it's quite rare. And so there are actually a lot of traditionally human pathogens, for example, Giardia, which was the protozoa that Ali brought up earlier, that can also affect pets, but then there are tons of others that only affect humans. And a good example of that is a close cousin of salmonella that actually causes typhoid fever.
Any other questions? Yeah.
Just upon kind of contamination between meat and vegetables, since it sounds like the bacteria requires some sort of animal cellular level to thrive, how long can it actually exist on vegetables?
It's a good question. So the question is, I mentioned that there is cross-contamination sometimes between animals' intestines and onto other types of food like vegetables, and he asked, what is the lifetime of salmonella on those other surfaces? And I actually don't know how long they live.
So the thing about salmonella is it can grow inside your body, and that's where it's happiest, but it also can grow outside, like we grow it on petri dishes in the lab. So it would really depend on the vegetable, so basically what nutrients the vegetable has on it or inside it and how much salmonella likes them. So one example, this is more common with E. coli than salmonella, but in alfalfa sprouts, the conditions where they take alfalfa seeds and sprout them are really good for a lot of bacteria to grow, which is why you see a lot of outbreaks from sprouts. So it's very dependent, but yes, it can grow on vegetables.
Thanks, Greg. Any other questions? Okay, if not, I'm going to pass off to Brandon, who's going to be telling you about the beginning of salmonella's journey into our body.
Is this okay? Can everyone hear me well? Just shout if you can't hear. How does this work?
Just clip it onto your body.
Quick intermission to transfer mics. We have three.
There are a lot of mics going on here.
One for this, one for the live.
Sorry. Okay, do you mind holding this?
Yeah.
Clip on. Okay. One. I don't have enough pockets. Okay. All right, okay. So everyone can hear me well, right? Okay, cool. Well, as Ali mentioned, I'm Brandon, and I'll be taking over from her to really go over this next section of the talk, where we're gonna really follow Salmonella that has now been eaten by a human and is now in the mouth, and we're gonna follow it as it moseys its way down into our intestines and finds a home there. So before we really start in the mouth and ask what's happening to Salmonella in the mouth, I just wanted to talk about a question that some of you might have, which is really, how many Salmonella do we have to eat in order to cause disease? And I want to just open it up right now and ask, does anyone have like a guess as to how many bacteria we have to eat, how many Salmonella we have to take in order for disease to happen in us?
1,000.
1,000? Okay, that's a good guess. Yeah.
Me?
Yeah.
506,000 million.
506,000 million. Okay, well, you're not far off. I mean, neither of you are far off. So the answer is really, we're not quite sure. So it appears that from the various studies that have been done on really how many Salmonella it takes, or really what is the infectious dose of Salmonella, that it seems to vary between from about 1,000 bacteria to about 1 million. Now this might sound like a very, very big range. So 1 million is about, well, it's exactly 1,000 times more than 1,000. So really, like why is this? And it's interesting because studies that give us this number of 1,000, they actually come from the public health agencies when they go back and do the math on real world outbreaks and try and figure out how much each infected person actually ate, and they come up with around 1,000. So you might be wondering where this million count comes from then, and this actually comes from very carefully controlled studies where scientists have gotten human volunteers to actually eat a certain amount of Salmonella and then monitor whether they got sick. And it somehow seems like in these kinds of studies, we get higher numbers required, and we're not really quite sure of this discrepancy. But either way, in any case, even this lower estimate is still quite a bit higher than, say, 1 or 10 bacteria. And we're going to see throughout my section of the talk really why we have to eat so many bacteria in order to cause disease. And just before we get back to the mouth, I kind of want to go back to something that Ali brought up in her section, and that's really, what do microbes or what do cells want? And she brought up this great quote from Jacob that the dream of every cell is to become 2 cells, and that's really to replicate and then to become 2 more of itself. So since we're talking about Salmonella today, I thought I would rephrase this in the context of our favorite bacteria and really state that the dream of every Salmonella is to become 2 Salmonella. So I've taken a little creative liberty with Jacob's quote here. And really, what do I mean when I say, like, what does this mean? The dream of every Salmonella is to become 2 Salmonella. It really means that when they're in our body, the bacteria are looking for 2 things, and that's food and shelter. Pretty similar to us. In order to become 2 humans, we're going to need food and we're going to need shelter. So when I do want to mention, though, that when I say food, I don't mean human food. So when I talk about food, I really mean nutrients and complex molecules that have been broken down into their simplest parts that the bacteria are able to see in their environment and to take up into themselves in order to fuel their replication. And so with this sort of framework of these 2 basic needs that the bacteria have in order to establish their home in our body, we're going to go through some different locations in the body and really see whether they satisfy these 2 criteria or not. Okay, so let's start really at the entry point of all Salmonella into the body, regardless of how you got it, whether you ate contaminated food, whether you shook hands with someone who really has bad toilet hygiene. All bacteria is going to be coming into your mouth. So the mouth is obviously not only the entry point for all bacteria, but it's also the entry point for all the food that you eat. And when you're eating food, you're chewing, you're spitting, sometimes they throw up, things are not going all the way in all the time, right? So the mouth is in constant motion. And really the direction that things are going is not the same all the time. It's a very diverse environment, it's changing all the time. And one really, really important aspect of the environment in your mouth is that saliva is actually very harmful to bacteria. So if Salmonella want to really stick around in the mouth and replicate in the mouth, they're actually going to have a hard time because the entire surface of your mouth is coated in saliva. So really, in the mouth, food has not been broken down enough for Salmonella to see it and to be able to access these nutrients. And because of the harmful saliva, they don't have shelter either. So the mouth really is not a hospitable place for Salmonella to live. So once Salmonella have really decided that they're not going to stick around in the mouth, the next point is to go all the way down the esophagus. And really, they next find themselves in our stomach. So the stomach, as you might remember from high school biology, is really where the majority of food digestion starts. So it's a very, very acidic location. So it's basically a sack of acid that just starts attacking anything you're swallowing down your esophagus. And when I say acidic, I mean about 10 times as acidic as vinegar or lemons. So it is a very, very harsh environment. And so this harsh environment really starts breaking down your food at a chemical level and will open up nutrients that the bacteria can begin to see and access. But at the same time, this acidity is actually very, very bad for bacteria and any living cell that you might happen to ingest. So again, even though there might be food here, it's certainly not finding any shelter. And really, once Salmonella has sort of gone through the stomach, it's not a good place to live. Really, what it hits is the intestine. And in order to sort of unpack why the intestine is such a great place for Salmonella to live and to call home, I just wanted to go over some brief intestinal biology and some basic anatomy so we're all oriented in the right direction. And so this will look pretty familiar. So this is the stomach here, and basically once food or bacteria has gone through the stomach, it exits into the tube that we call our small intestine, and basically food is starting to be pushed through this tube by muscles on the outside that are contracting in one direction. And during its journey through the small intestine, food is going to be further digested and further broken down, and inside the small intestine is where really the bulk of the nutrients of the food are going to be absorbed into your body. So this is where stuff like proteins and fats and vitamins and minerals are going to be absorbed into your body. And so once food passes through your small intestine, it exits into your large intestine or colon. And it begins its journey up here and across here. So in the colon, the main function of passing through food through the colon is to reabsorb any water that's left in the food so your body can use the water. And so your food passes through here, getting the water sucked out of it. And I think we all know what happens when it gets to the anus, so I'm not going to go over that. Okay, so now that we've sort of got a brief orientation of where we are in the intestines, I wanted to really just take a small slice of this tube, so a small slice of the large intestine, and talk about the layers of cells that make up the intestinal wall. So just to orient ourselves a little better here, there are two sides really to this wall. And that would be the side that the food, or what once used to be your food, is passing through on. So this is what we call the intestinal lumen, which is basically the open space inside your intestine where solid matter passes through. And on the other side of the lumen, we have basically where the blood vessels are, and where the blood is coming through and picking up nutrients that are being absorbed by these cells here and coming across into your blood. And so this is really nutrient rich, and it's feeding the rest of your body. So you can see here there are several really distinct layers of stuff. And so what are these layers? So these sort of pinky and white layers here, they're various layers of muscle cells, and these are basically the cells that are helping to contract and push your food through your intestine as it's getting digested. And this sort of darker pink band here, where there's a lot of purple dots, and the purple dots are basically cells. This band here, it's called the lamina propria, and it's basically a loose network of cells that contains a whole bunch of different kinds of cells, but as we'll see, some of the most important types of cells that are contained in this lamina propria are actually cells of your immune system, and we'll see how they play a role in your gut in a few slides. And right on top here is perhaps the most important layer of cells that we're talking about right now, which is your epithelial cells. So your epithelial cells are essentially the outermost layer of your intestines. So they're the layer that's actually touching the food and is actually accessing the nutrients that are being released from your food. And together, the entire layer of epithelial cells is called the epithelium. So so your epithelial cells are the ones that are actually taking in the nutrients, passing them through here, and then eventually passing them through the blood. And if we now zoom back out to our original question of, is the intestine a good place for Salmonella to live, we can again frame it in the context of those two criteria, food and shelter. So in the intestines, food is finally being broken down to its most basic components, and these are great for bacteria to access and to use to fuel their replication. And as Ali mentioned in her section, other microbes and other bacteria are actually flourishing in this environment as well. So we know it's a really good place for bacteria to grow. So in the intestines, we've actually satisfied both criteria of both food and shelter, making it a great place for Salmonella to live. And I want to just come back to this original question that we had of how many Salmonella does it take? I still don't have an answer to this question, so we're still not quite sure. But really, with respect to why the minimum seems to be about 1,000, we can now kind of think about it in terms of how Salmonella is making its way down to our intestines or to our gut. So imagine we start off with 1,000 or 6 Salmonella, and we eat this on, say, a spoiled egg salad sandwich. The first thing they see is the mouth, and so maybe some proportion of the Salmonella and some percent of the Salmonella is going to be, say, physically broken by your teeth, and they die, and then some don't make it into the esophagus, so their population is actually decreased by a fair amount. And this fair amount, once they bypass the mouth, they now have to face the stomach. And in the stomach, they have to face all those stomach acids that I just told you about. And this actually will also kill a very high amount of the Salmonella that reach it. So by the time really the bacteria have exited the stomach, there's only a few of them left. So even if we start with 1,000 bacteria, we might not be left with that many that actually successfully make it all the way down into our intestines. And so the mouth and the stomach really form what researchers term barriers to infection. So these are sort of aspects of our body that are actively or passively blocking infection by pathogens. And it's helpful to remember that this is really only to get to the intestine. So essentially, the rest of our talk is going to focus on what Salmonella is doing in the intestine. And before we dive in and look a little bit more closely at what Salmonella is seeing in the intestine, I just wanted to take a quick question break, if anybody has any questions.
So what's so harmful in our saliva that kills the Salmonella? And if I chew my food longer, am I more risk of getting a Salmonella?
Okay, so those are interesting questions. So the first question was, so what's in your saliva that is really like attacking the Salmonella? So there's basically a very potent protein called amylase. No, not amylase. It's lysozyme. Yeah. So there's a very important protein called lysozyme that basically can actually attack the bacterial cell wall. So the covering of the bacterial cell, so it can actually attack and degrade bacterial cell walls. So it's very good at basically popping open bacteria. And so the second question is really, if I chew my food long enough, will I be able to reduce my chance of getting a Salmonella infection? And so I can't give you a definitive answer on that, but I don't think the proportion really of a salmonella that you're going to be able to break by chewing is going to be like significant. And so if you if you're trying to chew your food for say an hour before swallowing it in the hopes of destroying bacteria, I'd say your priorities are probably a little mixed up. But yeah, I think the proportion that is destroyed by chewing is much less than is say destroyed by lysozyme acting in your saliva. So any other questions? Yes.
So you mentioned that like a bunch of the salmonella are going to die because of the saliva and chewing, and
right.
a bunch of them are going to die in the stomach.
Right.
How do they even survive at all? Like, are there special characteristics of the ones that do survive that allow them to get to this?
Yeah. So in the case of say, so the question is really like why are any able to survive really, or it's like why do some die and why do some live? Yeah. So basically, Salmonella is an imperfect bacteria, and basically it does have, say, in the specific case of stomach acid, Salmonella does have specific systems that it can turn on to evade really the acidity of the stomach. But in a population of Salmonella, some might be faster and some might be slower at turning on these systems. So if you're part of the slower part of your population that is turning on the system, you have a greater chance of being killed. And if you're one of the Salmonella that can immediately get down to the stomach and then immediately turn on your acid response system, then you have a higher chance of getting protected. So that means like it's not just a simple on and off switch. It's really a long, say, it's a bit of a gradient in terms of how they're able to respond to different barriers. Yeah. Yep.
The 1 million, like the study that found 1 million, was it did they know that they were always going to get infected, or did they do you know anything about how that study worked out? Like, was it a blind study? Because potentially, what if you just harden yourself that you're always going to get food poisoning, and then that means that you're less likely to get food poisoning?
Sorry, do you mind repeating that?
I was wondering if the 1 million number, if the volunteers were always going to be getting doses of Salmonella, because if they thought that there was always the potential of getting Salmonella, maybe they're having their bodies ready for they're able to deal with so much more than the unsuspecting Chipotle.
Okay, so the question is really so the question is with respect to sort of the infectious dose volunteer studies, how are those done really? So there hasn't been one done for I'd say 15 or 20 years. And I think the way from briefly going over these studies when I was preparing for this presentation, I found basically they're always feeding you bacteria. There's just how much bacteria they're feeding you. And they basically, between studies, they can also change which food they're feeding it to you. So I've seen it, sometimes they use yogurt, sometimes they use ice cream, sometimes they use meat, sometimes they just make them drink it. So I think depending on which food you're actually ingesting or taking in your bacteria on can affect how your disease turns out as well.
Thank you.
Yep.
Why do the bacteria in your mouth not get killed by lysozyme?
That's a good question. I don't. So one other sort of aspect to this is that there are also bacteria that are able to live in your mouth. And Katie's question is, why are they not killed by lysozyme? And I don't have
They have resistance mechanisms to them. So it's basically Salmonella hasn't evolved to grow in your mouth because it just randomly evolved to grow in your intestine. But some bacteria have randomly developed, say, really thick outer barriers around their walls that prevent the lysozyme from getting to them. And those bacteria, when they happen to get into the mouth, do better. And it's kind of just this random, what we biologists would call selection. And it ends up with some bacteria that are just really good at living there because they have different mechanisms to prevent lysozyme and other things in the mouth from killing them. It's the best answer that we can give.
So there are bacteria that have chosen to live in the mouth, and they are very different from Salmonella. So In terms of choosing a habitat over time, they become adapted to the habitat that is our mouth. And Salmonella has become adapted to the habitat that is our intestine. So, any other questions before I go on?
I think we should probably move on.
Okay, yeah. Okay, there'll be another question break in a couple minutes. So when we last left Salmonella, they had just exited our stomach and they're now in our intestine. And they've satisfied their 2 criteria of food and shelter. And really, like, what is the situation now? What are they seeing? If we think of really what the Salmonella are seeing, we can really build this situation bit by bit until we've sort of got a complete picture of the intestine. And so first we have our Salmonella. They're fresh out of the stomach and they're following the flow in the intestinal lumen, which is the open space inside our intestines. And they're just following the flow of the intestine. And surrounding the lumen are normal gut cells. So you might recognize this picture from a few slides ago. So this would be the lining of our intestine, and this would be here the epithelium, composed of epithelial cells. And for simplicity's sake, I'll just replace it with these cartoon cells here. So Salmonella is really seeing the normal gut cells, and they're flowing along through the lumen. So really, they don't want to keep going with the flow. So the flow is really strong in the lumen, and Salmonella wants to stay around in your intestine, and they want to stick around and replicate. So really, the goal is actually, once they've arrived in the lumen, it's to get closer to these epithelial cells where the flow isn't as great and maybe there are more nutrients. And really, if these were the only 2 things in the gut, this would be pretty easy for them to do. So as Ali said, Salmonella have special appendages called flagella, and they can use those to swim in a certain direction. So they could easily swim over to the epithelial cells. But there are actually several things in their way. And the first of this is actually a physical barrier in the gut that's called the mucus layer. So the mucus layer is basically a very thick, slimy substance that is produced by your epithelial cells, and it's basically a slimy surface that covers this whole layer and prevents anything from actually getting through, anything big getting through to the epithelial cells. And it's actually very hard for a bacteria to just simply swim through the mucus and get towards the epithelial cells. So that's a very, very tough, very physical barrier to these bugs. And instead of physical barriers, in addition to physical barriers, our intestine also has cellular barriers that Salmonella must face in their journey towards disease. And here I've represented these sort of immune cell defenses as these purple blotches called macrophages. So macrophages are basically very specialized immune cells that we can think of as rover cells. So they basically move around your intestinal epithelium constantly, and they're always reaching out and grabbing bits of your lumen and things that are passing by in your lumen and are near the epithelium. And they're constantly sampling these things and sampling their environment to ask whether the things that they're sampling are things that are dangerous or not. And so they have very complex ways of sensing whether something is a pathogen or whether something is beneficial or whether something is neutral or not. And if they do sense a pathogen, they're actually able to activate even more immune defenses. So these macrophages, we can kind of think of them like sentinels of the immune system. So Salmonella also has to evade these sentinels. And finally, in addition to these human cells that are in the gut, there are other bacteria in the gut. So represented by these diverse colored pills here. These other bacteria are really a collection of species of bacteria that we call the microbiome or the microbiota. And they're essentially the good bacteria that live in your gut, that are already in your gut. They're taking up nutrients and they're living happy lives in the gut. And so really, to get to here, Salmonella has to pass all these barriers. And if we think about this journey now, it looks a little bit harder than before when all Salmonella had to do was just swim to the epithelium. And so just to wrap up this section, I want to really focus in on this point here about other bacteria. And it's interesting thinking about why the presence of other bacteria that aren't causing disease is actually fighting the ability of a pathogen, say Salmonella, to cause disease. And so this question is sort of like, how do our resident microbes actually help defend against these diverse species of pathogens? And it's helpful to think of, again, those two criteria that all cells need. And that would be food and shelter. And while in the gut, these resources are plentiful and they're common, at the same time, they're not plentiful enough. And in a sense, they're actually limited because the bacteria can only, say, grow on so much surface of your intestine before there's no more room. And the food is not constantly coming into the intestine. Sometimes there will be shortages, and sometimes there will be periods where the bacteria are actually starving. And so if you think of what the bacteria are trying to get, if you have a group of, say, pathogens like Salmonella that are trying to access this, these nutrients and this shelter, but at the same time there's trillions of beneficial bacteria that are already in your gut and already using these nutrients and already taking up this space, it's actually very, very hard for a pathogen such as Salmonella to establish a foothold in this environment. Right, so to just really summarize my section, we sort of gone over the fact that pathogens are usually just looking for food or nutrients and a nice sheltered home to settle down in and make more bacteria. And while we found that our gut is a really, really great place for pathogens and bacteria to live, it's actually got a lot of defenses against these bacteria. And we'll actually see in a few minutes in Greg's section how Salmonella is able to counter these defenses. So I'd be happy to take questions about my section now. Yep.
How do cells like macrophages differentiate between Salmonella and your quote-unquote good bacteria?
Right. Okay. So the question is how do okay, wrong way. Okay. So how do cells like so the question is how do cells like macrophages tell the difference between good and bad bacteria? And really it's still an emerging field of research, so I would say the answer is not exactly known yet, but essentially the way that macrophages are able to tell whether things are good and bad is by sort of sucking in a little sample of what's outside and then looking at the molecules that are covering that cell's surface to tell whether they're good or bad. And basically, the difference between Salmonella and some of these quote-unquote good bacteria is that they have different sets of molecules covering their surface, and these are recognized in different ways by macrophages.
But, I mean, they're all bacteria. Wouldn't they all be considered foreign to the macrophage?
Right. So that's a really good question. So the question is, if they're all bacteria, why aren't they all considered to be foreign? And so this is a very still sort of researchers are still sort of feeling their way around this question. But the consensus seems to be that these bacteria here that are beneficial are actually recognized as a little bit foreign, and they have your immune system activated to just a little bit, but not a lot. And the reason why they're activating only a little bit of your immune system and not enough to cause, say, an entire immune response is still unknown at this point.
If you don't mind me jumping.
Yeah, sure, go ahead.
He's totally right in that the answers are not definitive in the field yet. But there are 2 points that I'll bring up. One is that the microbiome that colonizes your gut, it kind of starts colonizing you from birth. So while your immune system is developing during infancy and during your early years, your immune system is actually training on those bacteria and figuring out, okay, these are foreign, they're not human cells, but they don't seem to be doing us any harm, so we'll kind of let them hang around. When Salmonella comes in, and this is getting to my second point, Salmonella is unique in that it actually tries to traverse all these different barriers and try and access those cells, and that sets off a ton of alarms, not only with the macrophages, but a lot of other immune cells. And so it's partially about familiarity and partially about proximity and location. But
totally not a complete picture yet.
Yep. Other questions? Okay, cool. Then I will pass the torch to Greg. What?
An intermission.
Or, okay.
Great if you'd use the restrooms and get more snacks, and then we'll come back for the last part.
Yeah, take all my mics.
So, as Brandon initially went over, Salmonella has a bit of a dilemma when it gets to the intestines. I mean, there is food, there is shelter, but it's still kind of a hard place to live due to things like liquid flow, which is still very strong, especially when you're outside all those layers, physical barriers like the mucus, immune responses, and other bacteria. So Salmonella has to find some sort of solution to all these problems. And that solution, which can all kill by Salmonella, I forgot to edit that. And all of these things can be solved by Salmonella going inside of the actual epithelial cells, which is what we call intracellular. And this offers distinct advantages in that there are lots of nutrients because cells have to take in nutrients in order to feed themselves, and epithelial cells take in a lot of nutrients to feed the rest of the body. You can avoid the immune response. Most immune responses are trained outside human cells because that's where most pathogens are. Once you get inside, they're a lot more sparse. There are a lot fewer of them. And there are obviously not supposed to be any other bacteria living inside your cells. You have a bit of a problem if you have like multiple species of bacteria inside your epithelium. You should get that checked out. Anyway, so the first step in getting inside of human cells is to get to the human cells. And Ali mentioned flagella earlier, and they mentioned how they might not be strong enough, but it turns out that Salmonella have really strong flagella. So I'm just going to go into detail on how these flagella work. So if you see in all of our diagrams, we have all these little spindly fibers attached, which are the flagella. And what happens is all these fibers collect at one end of the cell. So they combine in their strength. And then these fibers all rotate together in a kind of corkscrew motion, and they can actually propel the cell forward with a ton of force. And this causes bacteria like Salmonella, which are on the outside of this whole layer of bacteria and mucus, to propel right through all of it and reach the edge of the epithelium. So it's not a very exciting response in that you already know that Salmonella can swim, but it turns out they can swim really, really fast and really, really strong. The second step, before I go into the second step, I have a video. So this is I've gone over how bacteria swim, but because a video can tell, with pictures 1,000 words, a video is like 1,000,000, right? So here is a video of bacteria swimming. And you can actually, if you look for a little bit of detail, these are individual bacterial cells. There's some obstacles. This is on a microscope slide. There's just a little bit of stuff in the way, which they're actually navigating around. And what's really interesting, if you look at one individual cell, it's kind of vibrating as it goes forward, and that's because the flagella that are collecting behind it are spinning really, really fast and causing the body of the cell, which is all that we can see because flagella are too tiny to see, causing the body of the cell to vibrate as it goes on. And if you also notice that these bacteria are going very fast relative to the size of their bodies. Like that guy just zoomed down to the bottom of the screen. So these are very powerful motors, and that's how Salmonella can get through these layers of bacteria and these layers of mucus to reach the epithelium. So I've gone over how Salmonella gets to the epithelial barrier, but the process of getting inside of a human cell requires another step, which is getting really close to the human cell. It wants to kind of become its friend, convince it that it's a great idea to let it in. So to do this, Salmonella uses other appendages that are similar to flagella but different structure on the outside, called fimbriae, which are long, spindly fibers with very sticky ends. And they actually, when you use a very high-powered microscope to look at them, they kind of look like this. So here is a bacterial cell at the center, like Salmonella, and here are a ton of fimbriae jutting off. And you notice that there are a lot of them. There are actually a lot more fimbriae than there are flagella when the Salmonella is approaching the outside edge of the epithelium. And I added these fimbriae to this diagram just so you can observe that every fimbriae that is at the surface of the epithelium sticks to certain molecules on the cell and allows the Salmonella to get really, really close. So the final step of the process of getting into a human cell, I like to call get eaten. And getting eaten involves having this cell, which normally epithelial cells don't absorb bacteria. They absorb nutrients, they absorb small little molecules to bring them into your bloodstream, but anything as large as a bacterium is not normal. Normally they don't do that, so the Salmonella has to convince them to do that. In fact, it has to convince these epithelial cells to kind of act like immune cells, like macrophages that Brandon mentioned earlier, and eat the Salmonella. So kind of a little diagram of how this might work is you have your epithelial cell, your Salmonella that is adhered very closely, and the next step is the epithelium kind of envelops it with its outside membrane or outside edge. Oh, the mouse is on the edge. Thank you. But anyway, so the next step would be to envelop this Salmonella bacterium. But again, as I mentioned, these epithelial cells don't normally do this. So Salmonella has to find a way to convince the epithelial cells to do this. And it does that by changing their behavior. It actually takes these specialized molecules that I'm going to call molecular remodeling tools because I think it sounds cool. And Salmonella is going to produce these molecular remodeling tools and push them into the epithelial cell, which will actually cause the structure of the cell to change and cause the outside edge of the epithelium to eat the bacterium. But this poses a bit of a problem because these remodeling tools are made inside the Salmonella, and they have to remodel the human cell, which is not the Salmonella. So somehow this bacteria on the outside of the epithelium has to send these little molecules into the epithelial cell. And this is, I think, one of the coolest parts of Salmonella biology, or even bacteria as a whole. I'm a bit biased. But it uses this special structure called a type 3 secretion system, which is a lot of words, a lot of jargon. We just like to call it the T3SS. And it essentially acts as a molecular needle. And if you look at this cartoon of one right here, it almost does look like a needle. It has this very wide part where you put the initial, in this case, the molecular remodeling tools. For a needle, it would be whatever you're injecting. And it pushes it through across the intestinal lumen, there's still a little bit in between, into this human cell. And so it delivers remodeling tools, and these molecules are the ones that actually cause the epithelial cell to envelop the Salmonella. And what's especially interesting about the type 3 secretion system is you can actually, using certain methods, separate type 3 secretion systems from Salmonella or other bacterial cells and look at them under extremely high-powered microscopy. So this is a cartoon that somebody drew up with Photoshop. This is an actual microscopy image of these individual molecular needles. So you can see that they actually do have this very wide base and this long tip that can push into the human cell. And so the summary of what happens is the Salmonella will get close, adhere, and use these type 3 secretion systems, represented here by these small red shapes, to inject molecular remodeling tools into the epithelial cell. These will cause the epithelial cell to change its behavior and start spreading this little enveloping package. And the Salmonella will then get enveloped and go into this little pocket inside the epithelium. But these are, again, all cartoons, and I think, as I said before, a picture tells 1,000 words. So we're going to get really up close and personal with this, using, again, high-powered microscopy. So to start out, this is something we call electron microscopy. And this is a human cell underneath the microscope. And here's a Salmonella. And you're actually viewing it, it's a slice, and you're viewing it on its end. So it looks like a circle. The cell is actually shaped like a rod, but you don't always choose how you're looking at it. So it's on the surface of this human cell, adhered really close. You don't see much space in between them. The next step is it has injected these factors. The type 3 secretion is not being picked up by this microscope, but you just trust that it's injecting these factors. You can see these two little arms coming out and starting to envelop the Salmonella cell. And then the next step, it completely gets encapsulated by the human cell. So this is actually kind of a complete story. It goes from outside, envelopment, and then inside this human cell. But even cooler than looking at electron microscopy pictures of Salmonella is looking at a video. And this is, again, one of the coolest things that you will ever see in all of science, I can guarantee. This is Salmonella, and the Salmonella here are represented by these green blobs because they actually got it to express a bright green molecule, getting eaten by a human cell that you can see here. And we will start this video. It's not very long, but it is really cool. And bam. So let's go into a little more detail on that. So we start out, and we have a Salmonella, which is On the outside, and I'll just move the cursor for now. On the outside edge, it's very closely tight to the cell, just as we saw in our cartoons and in the previous images. And the cell is looking pretty normal. It's an epithelial cell. It doesn't really want to envelop a salmonella. But the salmonella is going to change that. Injects its remodeling factors. Oh, I was meant to pause it there. Okay, let's just start it again. Okay, and this is kind of mid-encapsulant. So you can see there are these very long strings that are being pushed out by the cell. That is the type 3 secretion system remodeling happening. And the salmonella has started to get enveloped into this human cell inside a petri dish. And then, finally, the human cell envelops the salmonella and the surface starts to return to normal. So this is a very quick, very it's different. The cell doesn't normally do this. It wants to return to normal, and the salmonella doesn't want to disturb the cell too much. So after it's enveloped, the salmonella is totally fine with the cell returning to a nice normal surface. And you know what, just because I really love this video and it's so cool, I'm going to play it again. Boom, okay. How long does that take? So overall, this is a sped up image. I wish I remember from the paper how long the time scale was. I think it's a matter of minutes. Yeah. They're the ones who look at these much more often than I do. I work with a much slower bacterium. So this happens in a matter of minutes. And we'll move on to the next slide. Okay, so we've now established Salmonella is inside the cell. It's a nice little happy home, it's a little pocket. And you can almost look at this if you strike back to this quote that Brandon and Ali both used, that the dream of every cell is to become two cells. And this is what Salmonella has been trying to do the entire time. I am going to note that I used Francois Jacob's quote because he has a Nobel Prize and Brandon does not. So I think he deserves to be on this slide. So the dream of every cell is to become two cells. So Salmonella, because what bacteria do is they divide in half and grow, can actually become two cells now that it's in this nice happy environment, no flow of the intestine, lots of nutrients, less immune response. It can become two cells, become four cells, and you can almost say that Salmonella is living the dream. So, it's not tacky, I swear. So, we've reached this stage, and Salmonella is living happily inside your cells, it's replicating, it's gotten to its nice home, but if you remember that Salmonella also induces this thing called diarrhea. So how does it do that? So first we're going to look into why it wants to do that, and the reason why Salmonella causes diarrhea is because it wants to move on to a new victim, and regular bowel movements just aren't fast enough. Because if the goal of a Salmonella is to become two cells, it wants to become two cells, but in more places. Because one human just isn't enough. You can't grow enough for the appetite of this bacteria. So Salmonella spreads via diarrhea, as we've already mentioned many times in this talk. And this diarrhea is caused by inflammation. And what inflammation is a type of immune response. So Salmonella spends a lot of its life trying to avoid immunity, trying to avoid macrophages and if you know anything about antibodies or something, like these different words you might have heard of, all have to do with the immune system. And Salmonella normally doesn't like it, because the goal of immunity is to kill pathogens. But in this case, Salmonella kind of turns the body on itself and takes advantage. So when Salmonella is inside this cell, and there's still some Salmonella bound on the outside of this image, it's going to send immune signals down into the lamina propria and the muscle cells in the blood, just like Brandon mentioned. And the macrophages and other cells there are going to pick up those signals. But the signals that Salmonella sends aren't complete. They're just enough that this is going to cause some low level of inflammation. And one kind of hallmark of inflammation is liquid flow. Because when the body sees, say, a pathogen in the intestine, its first response is to flush it out. So the body sends a ton of liquid from below this level, both from the cells and through the various gaps, and tries to push out anything in the intestine that might be causing it problems. And this actually overwhelms the capability of the large intestine to absorb back all of the water, which causes diarrhea. And what happens to Salmonella in diarrhea is that any cells that are still on the outside are going to be pushed off by the liquid flow. And one really interesting thing is we actually don't know what happens to the cells inside the epithelium at this point. It's still a very active area of research. They might also lyse or break open these cells and escape with the liquid flow to enter the diarrhea. But either way, they are very important in causing these immune signals and causing this liquid flow to spread Salmonella to its next unhappy victim. So I've mentioned how Salmonella gets into the intestine and how it lives there happily and how it causes diarrhea to spread. But another thing that Ali mentioned way back in the beginning of the talk is that Salmonella infection normally only lasts about a week for a healthy adult. So all of these things I'm talking about has been Salmonella taking advantage of the human body and using it as its home, but the human body does eventually fight back. And I like to call that tipping the balance of immunity. So this is all with the goal of clearance. Clearance is the definition of clearance is when a host, or a human in this case, gets rid of all of the cells or particles of a pathogen that's within it. So clearance in terms of Salmonella would be the human body getting rid of all of the Salmonella. And in the beginning, when Salmonella is being happily infected, you have your pro-human immune responses and your pro-Salmonella. And as I just described, Salmonella is taking advantage of the human body. It's tipping the responses to be more pro-Salmonella than pro-human. But the human body has been around for a long time. We've evolved some very, very complex responses, so what ends up happening is we actually kill the bacteria. So human cells, even though Salmonella is not that exposed to most immune responses inside the cell, there are still some ways that we can deal with it, including, say, acid killing. There's actually a really cool system where human cells can inject acid into bacteria that are inside them. There's more immune sensing, not as much as on the outside of a cell, but there still are some ways that the immune system can detect bacterium inside a human cell. Cells can actually self-destruct. They can kill themselves and kind of sacrifice for the greater good if they see that there's bacteria inside them, or they can call for help via a variety of different ways. Basically, they send certain molecules out, and that brings more macrophages, more immune cells, to the site of the Salmonella's entry into the human cell. So all of this results in a tipping of the balance of immunity. So whereas pro-human was high up and not very effective before, it's now much heavier. It has tipped the balance against the pro-Salmonella immunity. And this eventually does cause clearance of the Salmonella. Things like bacteria, immune sensing and acid killing and all those other things that can cause bacterial death wipe out all of the Salmonella in the intestine. And in a healthy adult, this will happen within a week. So just to summarize what I went through, Salmonella uses special methods to travel and get inside human cells, including flagella, which allow it to power through very tough physical and bacterial barriers, fimbriae, which allow it to adhere very tightly to the outside of epithelial cells, and the type 3 secretion system, which allows it to inject molecular remodeling factors to get enveloped by the human cell. Inflammation then causes diarrhea, which allows Salmonella to move on to a new victim, because Salmonella does want to spread and become more cells in more places. And finally, the body does eventually clear Salmonella by using effective anti-Salmonella immune responses. And at this point, I'd like to take questions on anything that I just said. We will have a general question session at the end where you can ask Ali and Brandon more too. But if there's anything that you guys want to ask about this part of the presentation, feel free to shoot. Go ahead.
If you're not healthy or it doesn't flush out in a week, what is done medically to help people who can't get rid of it on their own?
So the first thing that the question was, if there is no clearance, if people are not healthy enough, their immune systems are not good enough to flush out the bacteria, what can we do medically? What can doctors do to intervene? And the first answer is kind of like increase the amount of hydration. So for a lot of diarrheal diseases, once you get to the hospital, if it gets that bad, you can actually get an IV of fluids and electrolytes. Another step that you can take is antibiotics. There's a bit of disagreement in the Salmonella field about exactly what good antibiotics do, because by the time you are having massive diarrhea, the Salmonella have done a lot of their damage already. But there is Salmonella are bacteria, and just like most bacteria, they can be killed with antibiotic drugs. And there's probably other things that doctors have come up with beyond that, but those are really the basic two, is increased hydration and antibiotic treatment. Sure.
Right now there is this like a trend when if you're very sick, you go to the doctor, they don't give you anything because they say they can make resistant the infection to antibiotics. So my question is, if that is the case also, the Salmonella.
Yes, so resistance is a problem with pretty much every bacterium everywhere. And the question was, is there, because antibiotic resistance, the reason why doctors sometimes don't prescribe antibiotics if they aren't sure what's causing your disease, because antibiotic resistance is a problem where if you use the drugs too much, they stop working because the bacteria are no longer affected by them. And Salmonella, just like any other bacteria, can become resistant. I'm not actually sure of what the rates of resistance are in Salmonella, if any of you guys know. I would imagine they're lower than for many other bacteria because we don't use antibiotics as much, but the easy answer for a microbiologist is that there's always an issue with antibiotic resistance for any pathogen out there. What about you? You had a question?
How many Salmonella can enter the body at one time?
How many can enter the body at one time? Oh, well. So bacteria are, in the outside world, bacteria are very, they don't accumulate to that high levels. So you could take in billions of Salmonella at once maybe. But it gets to a point where, so bacteria, even though they're very small, once you get into the kind of trillions number, that's when you could actually see a lump of bacteria with your eyes, and that would be hard to consume. I'd say maybe, if you actually want an upper limit to the amount of Salmonella you could consume in one sitting, I would ballpark it, like 100 trillion maybe? But yeah, so the short answer is, in the real world, you're probably never going to get more than on the order of billions to maybe trillions of Salmonella at once, but you could, if you really wanted to, I don't know why you would, consume more than that. That would not end well though. We aren't supposed to give too many recommendations, but I would recommend very highly against that. Any other?
Where do the cellular remodelers go after injection?
After injection?
Degraded or?
Yes, so the question was, after these cellular remodeling factors are injected into the host cell and do their job and develop the Salmonella, what happens to them? And I'd say yes, that generally there are methods for breaking down proteins or molecules in all human cells, and there's no reason for Salmonella to make these proteins extremely resistant to getting broken down, because they don't need them anymore. Once you inject them, once the Salmonella is inside the cell, there's no need for these remodeling molecules, so they will get broken down, yes.
Is anyone looking at maybe co-opting that mechanism to deliver drugs?
Yes, that is, so the question was, has anybody looked at co-opting Salmonella's mechanism of getting inside cells to deliver drugs? And the answer is, for Salmonella, I'm not completely sure, but there are other bacteria that live inside human cells, and some of them do have mechanisms that have been co-opted to deliver different drugs, yes. I know I actually worked on a project about 6 months ago using a very specific factor that a different diarrheal pathogen, Listeria, uses, where it forms really big pores and membranes. And I looked at a method where you took really, say, E. coli that can't cause disease and put them into the human body, and then naturally some cells will eat them up, or you can express a factor like Salmonella invasive proteins, and once the cells get inside, they'll burst and release, say, new drugs, large drugs that normally couldn't get inside cells. So the short answer is yes. People are looking at how to use factors, maybe not specifically Salmonellas, but from bacteria like Salmonella to deliver drugs.
We're gonna have to move on.
Okay, we're going to move on. And again, if you have any more questions, there is another session at the end where you can ask Allie, Brandon, or me, and not rely on my answers. So, there we go. So we've talked for the last hour and a half now about Salmonella and how, what it is, what an infectious disease is, what the bacterium looks like, how it gets into our body, down into our intestines, and how it stays there and is eventually cleared. But what can we learn from this? And why do any of us care is a better question. Why do thousands of scientists study this disease? And the short first answer is that Salmonella is a really important pathogen. As Allie and Brandon went over, it causes millions and millions of cases per year and a lot of deaths. So if we do research on how Salmonella causes disease, we could find new ways to make drugs that target different parts of this process. So new treatments could reduce disease, so both reduce death from Salmonella and reduce the length of disease, or the intensity, so the amount of diarrhea that you have. So that is the first justification, the reason why a lot of us study it. The second is that a lot of bacteria use similar methods. So, for example, E. coli, which is another intestinal diarrheal pathogen, uses the type 3 secretion system and Fimbriae. And a lot of other different intestinal pathogens and other pathogens use similar mechanisms, including my favorite pathogen of all time, which is probably a terrible thing to say, but I love studying it, tuberculosis, which, as you can see from this old-timey French poster, has been a problem for a while and is a problem in a lot of different parts of the world. Also, when our cells fail, like they do in Salmonella, where they allow things to happen that they shouldn't, we can actually see how they work. So you can investigate, say, for Salmonella, you can look at, say, why do cells allow themselves to be remodeled? And when you look at how that happens, you can figure out how the cell is actually structured and how it forms that structure. So there are some really important lessons that we can learn about our own biology by looking at things like Salmonella and how it infects us. And finally, learning about how life works is really cool. It's just generally, biology is learning about this. It's the science of life, and we do love to study all parts of that. And we'd like to acknowledge everybody who helped us along the way, people who gave us things, people who kept us healthy, like our microbiota, and our sources of information in this thing. And we'd also, Science in the News, would like to thank these organizations for their generous support. And we also do have a lab tour going on after this presentation. It's going to be a walkthrough of the Walder Lab, as has already been mentioned, where all three of these lovely people work. And we are going to depart following these final questions. And there is a sign-up sheet, maybe. I don't know how that worked. We're figuring it out. Just meet, kind of coalesce down here near the podium after the presentation.
Yeah, let's take a few questions for the whole group, and then we'll kind of break off. And if you want to go on the tour, you can just meet down.
Yeah, so stick around, and we'll take you there. So, thank you very much for listening. And if you have any questions for any of us, now's the time to